Hello, my name is Yun Che, and I'm presenting on RT-qPCR and RNA-Seq analysis of SOD3 detox and redox. In this experiment, the genes of our interest are under the condition where oxidative stress is induced. And oxidative stress is often an underlying conditions of cancer, inflammation, and other diseases where the free radical species could damage our cells. Superoxide, peroxide, and hydroxyl radicals are all considered as reactive oxygen species. So, we would like to investigate how hydrogen peroxide could affect the gene expression in a human cell line, HeLa. It is well known that the expression of a gene often determines many cellular activities. And to enable or disable such activities in sensitive environment, gene expression must be tightly regulated. By doing so, homeostasis will be maintained and the body will remain responsive to many diseases such as cancer. As we see in the blue arrows, we first isolated the RNA, then RT-qPCR was performed. All 12 of our genes of interest were treated with 300 micromoles of hydrogen peroxide for 24 hours. Further analyses were done by Panther and BioBox. Here is the first result of the RNA isolation. The concentration of control sample was 363.8 nanograms per microliter and the purity was 2, and the RNA concentration of treatment sample was 272.9 nanograms per microliter and the purity was 2.02. Notice that the control sample was replaced with the provided RNA because the initial concentration was only 43.4 nanograms per microliter. The reason why I am emphasizing this result is because the quality of RNA sample is the most important step of the experiment. We have to have good quality and quantity of RNA to begin with, otherwise the RNA-qPCR or RT-qPCR results may not correspond with the RNA-seq results. In this table, we compare log 2-fold change values of RT-qPCR and RNA-seq results. Note that the highlighted GOI are the ones consistent throughout both of the experiments. Also, the positive values indicate upregulation, while negative values indicate downregulations. So here, the top 6 are upregulated and the bottom 6 are downregulated. This is a delta-delta CT graph which is used to determine the relative gene expression of RT-qPCR. Once again, the genes here with the arrows pointing are the consistent genes. We also notice that the PTGS2 has the highest magnitude while MAP kinase 1 has the smallest magnitude relatively. Here is the cellular component, molecular function, and biological process bar charts for downregulated genes. Binding and catalytic activity of the downregulated genes are observed from the molecular function graph. For the graph on the very right, the biological process with the highest hits were cellular process, metabolic process, and response to stimulus. Similarly, among the upregulated genes, binding and catalytic activity were found common for molecular function. And for the biological process, cellular process and response to stimulus were categorized as the process with the most hits. Now, comparing this with the downregulated genes, all 12 GOI appear to have some role in binding and catalytic activity and commonly characterized under cellular process and response to stimulus. Therefore, we can infer the regulation of these genes to have an effect on various cellular activities. Prostaglandin endoperoxide synthase 2 was upregulated in this experiment, and by its reactome pathway, it is suggested to secrete cytokines to regulate cellular activity via signal cascade. PTGS2 is responsible for enzymatic activity and production of prostaglandin, and it is involved in cell adhesion, apoptosis, resistance, and tumor angiogenesis. We can hypothesize its upregulation due to being under oxidative stress, where PTGS2 is upregulated to induce TNF and interleukin-1, which are both pro-inflammatory cytokines, to promote tumor angiogenesis in cancer cells. MAP kinase 1 was downregulated in this experiment. The reactome pathway suggests that MAP kinase 1 is responsible for the cascade of signal transduction from cell membrane to nucleus. MAP kinase 1 is known to mediate cell growth, proliferation, and differentiation, and it translates extracellular signals to intracellular response. We can hypothesize the downregulation of MAP kinase 1 as a response to oxidative stress. MAP kinase 1 is typically activated upon oxidative stress, so therefore downregulation of MAP kinase 1 provides inhibition of further cancer cell progression. Our assigned gene of interest, SOD3, was downregulated in this experiment. The reactome pathway suggests that cleaved SOD3 forms a tetramer which dictates the distribution of the protein in the extracellular space. Down below is the reactome pathway. First, cleaved SOD3 in Golgi lumen forms a tetramer in the lumen, where then it is formed in the extracellular region, and the SOD3 catalyzes the reaction of superoxide to hydrogen peroxide, and the hydrogen peroxide is further reduced to water by GPX3, 5, and 6. SOD3 being the antioxidant enzyme, it catalyzes superoxide radicals to hydrogen peroxide and oxygen. It is hypothesized to increase tumor response. Therefore, the downregulation of SOD3 suppresses tumor response. It is mediated by redox reaction of superoxide to hydrogen peroxide, and its inhibition leads to detoxification, which ultimately provides cell protection from oxidative stress. Here is the take-home message of the experiment. The regulation of gene expressions under oxidative stress were observed using RNA-Seq and RT-qPCR. SOD3 was hypothesized to be downregulated in response to oxidative stress. Downregulation of SOD3 turns off the catalytic activity of superoxide to hydrogen peroxide, which results in cell protection by reducing hydrogen peroxide concentration. MAP kinase 1 was downregulated to prevent cancer cell growth and proliferation induced by oxidative stress. PTGS2 was upregulated to induce TNF and interleukin-1, which are pro-inflammatory cytokines to promote tumor angiogenesis in cancer cells. Thank you.